So today I will be actually talking about an overview of gene expression. So there are certain things which you people should know because gene expression is there in your genetic syllabus and it is not given in a proper manner. Without having the basics of a gene expression, you cannot go into regulation of gene expression. Before making a house, you have to make a design of the house. Without making a design, you cannot randomly learn. You have to go to the first floor, to the second floor, to the third floor, to the fourth floor. You rise higher like this. You just cannot start from the fifth floor. So the syllabus actually is very poorly designed. It starts with the gene regulation, and there is no basics about what is gene expression, what is central dogma, what is anything. So I just today's class was basically to introduce you people to this part. You don't need to study in details, but at least you don't need the whole wide stairs to get up. Maybe if you get a little bit of space, you can at least reach up to the fifth floor. But this space, the other part of the space, it remains empty. In any case, you have to study it eventually. But to get to the fifth floor, at least you need to get to have a small, narrow basic where one person can pass. You don't need a wide area. If you have a wide area, then it's easy. You can go comfortably up. But here, uncomfortably, at least you can go and start understanding things on the top floor. So, with this, I'll just share my screen, and after what I'm going to talk about is the overview of gene expression, the first part. Okay, let's get back to the whiteboard. Is the blackboard visible? Those who are online.
Yes, sir.
Okay. So we will be talking about overview of gene expression. So these definitions are not the final definitions. There are different types of definitions which you people can will be able to study. Will be having to study later. But first, before gene expression, what is a gene? We have to define a very poor definition. It is not a thing. It's a working definition. I would say that how you can go ahead to study other things. But later, the definition of gene will change. It will enlarge. And then you will understand that the definition of gene is not so easy. But right now, as a working solution, I would say it's a segment of DNA. Then it should be coding for a codes, or it has got codes. Codes are present, and these are nucleotide codes. These are nucleotide codes which are present, and these codes, they are read as triplets. So what are these codes? They are A, adenine, T, G, C. These are the codes which are present in DNA. 4 new bases. It's just not bases. These bases are attached to a sugar molecule, ribose or deoxyribose. মা কলেজ গেছে মামা, ওই প্রতিমা পিসি আছে ওইদিকে। Deoxyribose sugar, and that is attached to either a single phosphate, then it is known as monophosphate, 2-phosphate, it is known as diphosphate, 3-phosphate is known as triphosphate. Adenosine triphosphate, guanosine triphosphate. If there is one phosphate group is attached, so this base is attached to a sugar and that is attached to phosphates. There can be 3 phosphates attached: monophosphate, diphosphate, triphosphates. So in a DNA strand, it is in a monophosphate form. They are attached. All the DNA are attached one by one. They are attached, right? So they are read as triplets. So triplets means it is read as, say, A, T, G. 3 nucleotides are read. Then G, C, A. Like this, it goes on the DNA. It's on the DNA. It is present in a linear sequence like this. So it is read as triplets, and it produces RNA. This RNA eventually may or RNA or this procedure RNA may or may not produce protein. Sometimes RNA can be the final product. In case of non-coding RNAs, they are basically especially non-coding RNAs. What do you mean by these non-coding RNAs? Most important is your ribosomal RNA. This RNA is present in the ribosome. Where what is ribosomes? What does ribosomes do? Synthesize mRNA. They don't synthesize mRNA. Ribosomes make proteins. They make polypeptide chains. Reading the mRNA code. So this process which I have explained, that a segment of DNA on which some certain specific codes are present, this code is present as triplets, and then these triplets, they are read by an enzyme, and this they make an RNA. This RNA eventually is made into proteins. From the RNA, proteins are made using ribosomes. This is the nutshell of gene expression. And now in gene expression, under different conditions, there is different types of gene expression. Under condition, there is different type of gene expression. Think of a bacteria. This bacteria is actually living. You have a medium where there is lots of glucose present. There is no lactose present. So why will the bacteria waste energy and make enzymes which metabolize lactose? It doesn't need. It has got glucose. It is happy on glucose. So these lactose genes are shut off. If the bacteria has got a medium where there is no lactose, it is a shut off. No required. When there is lactose in the media, only then the enzymes to metabolize lactose are needed. They need to take energy. Otherwise, क्या जरूरत है? I don't need it. Why हमको फालतू में क्यों बनाऊंगा? पैसा क्यों खर्च करूंगा? I don't need another laptop. Why will I waste another money? So that money is saved. Shut off. Spending is shut off. So this is how this expression is regulated. Suppose it's very hot. All the genes which are responsible to protect you when things are hot, they are expressed. And what is the final expression product? Protein, the functional enzyme or the chaperone. The protein is the functional product. Any gene, so whether it is insulin or whether it is a heat shock protein, it depends on the sequence of nucleotides which are present on the DNA. It gives a sense, and the intermediate phase is the RNA. On the DNA, the RNA code is made just complementary. Complementary of A is U or T normally U in RNA, G, C, it is like this. So complementary of A is T or U, and complementary of G is C. So whenever there is a C, the complementary opposite is G. T is absent in RNA. Instead of T, U is present. Uracil. Otherwise, everything is same. A, opposite of A is U or T. If it is in DNA, then it is T. If it is opposite in RNA, it is U. So, and in DNA, U is absent. T is present. Now we come to a very fundamental concept of gene expression, that is, let's do it like this. I can remove everything. I'm not liking this color, so I'll use another color. That is the concept of central dogma. You have to read this concept of central dogma. Keep in mind. This means you have a molecule of DNA. DNA is a double helix. This is DNA. This DNA, during cell division, this also replicates. It makes more copies of itself, and this process is known as replication. And then through the process of transcription, single-stranded RNA is formed. And through the process of translation, protein is formed. This is the functional product. Although sometimes, let me mention in star, sometimes RNA can also be a functional product. Sometimes RNA can also be a functional product. It can also function as an enzyme called ribosome, which cleaves phosphodiester bonds which hold DNA. Then there is another process. It was seen that this is all one directional. DNA is making RNA, RNA is making protein, but proteins cannot make RNA. This is the functional product. It was thought that RNA also cannot make DNA, but later to a process of reverse transcription. RNA can indeed make uh, DNA. Now, it was thought that RNA cannot replicate, but this was also found out. RNA replication, RNA dependent RNA polymerase, RNA replication. It was previously thought that only, not in all organisms, in certain organisms, it was seen that RNA, you don't need DNA to make RNA. First, we knew that through the process of transcription from DNA, you have to make an RNA through an enzyme known as RNA polymerase. This was the known concept, but this concept eventually, in our body doesn't. In certain viruses, it was found that RNA is good enough, that RNA only replicates by itself, and the enzyme is RNA dependent RNA polymerase. So this was a new discovery, but it is not very common. So this is basically the central dogma. Now we will talk about some differences in gene expression in prokaryotes and eukaryotes. Let's consider the case what happens in prokaryotes. This prokaryote is a bacteria. Let's say this is an E. coli. This is an E. coli cell. And in the nuclei, prokaryotic cell, you have seen there is no nucleus. All the DNA is organized in a nucleoid structure. This is the DNA organized as a nucleoid structure. Now, here, say in a section of DNA, transcription is taking place. Transcription is taking place. Messenger RNA, I am calling the term a messenger RNA or RNA, it is being formed. Right. At the same time, ribosomes bind on this. Right. Ribosomes bind. ribosomes bind and proteins are synthesized. So one thing you can see, that the moment that as transcription is taking place, translation also is starting. So in most important part is in prokaryotes, transcription and translation are closely coupled. There are other differences like in the chemical structure of the mRNA, which is present. No 5' cap is present, no 3' cap is present. There is no introns and splicing and all these things. But these things you will learn much later, maybe in some other semester. According to your brilliant semester syllabus, these things are not in this semester. But you have gene expression in this semester, which they could have easily avoided. Okay, this is the situation in prokaryotes, which we have seen. In eukaryotes, the system is a bit more complex. What happens in তোমরা এটা চাইলে ইউ ক্যান গেট দিস, মানে এই জিনিসটা তবে নিজের লেখাটা খুব ভালো। এটা কিছুই না, এটা একটু জাস্ট আই ক্যান মেক ইট স্মল। এই তো যা পড়িয়েছি সব আছে। এটা ইমেজ, আই ক্যান সেন্ড অ্যান ইমেজ। কিছুই না, অল আই হ্যাভ টু ডু ইজ গো হিয়ার। এখানেই আছে সামথিং অ্যাবাউট export as image, give it a name, say class, and it immediately I can save. It's saved. An image I can send through WhatsApp, you can see. But let me finish this first and then I will send you. এগুলো করা হয় না, রেকর্ডিংটাই থাকে। Okay, in eukaryotes situation is a bit more complex. One important thing is the presence of organs and organelles. In an eukaryotic cell there is organelles present. So this is the big cell, and inside the big cell there is a nucleus, and in the nucleus there are chromosomes. DNA is organized in chromosomes. It's not loosely present. They are packed, tightly packed with the help of protein. When transcription takes place, they become diffuse. So here, first through the process of transcription, pre-mRNA is made. Just pre-mRNA is made. Then what happens over here is processing takes place. Pre-mRNA processing takes place. Processing are, first is splicing. There are regions known as introns in particular in this, they are spliced out. Just know the names, you don't need to study the details. Splicing, then poly A tail addition. A is added at the end of the DNA. And one more thing, a 5' methyl guanosine cap is added. A methyl guanosine cap is added at the beginning. This is to protect the mRNA. So splicing happens, all introns are removed, it becomes much more smaller. So what you come actually, then you have the mRNA made. So all these processes, they take place in the nucleus. Then what happens? This mRNA is exported to the cytoplasm. Mhm. mRNA is exported to cytoplasm. In the cytoplasm, you have ribosomes now binding. Ribosomes bind and translation takes place. In cytoplasm, ribosomes bind and translation begins. It is also not simple. There are two types of ribosomes. One is endoplasmic ribosomes, endoplasmic reticulum ribosomes. And the other is cytosolic ribosomes. You have read this in cell biology, right? This thing, cell biology হয়েছিল না তোমাদের? Cytosolic ribosomes. So cytosolic ribosomes are free ribosomes. mRNA comes, it binds, translates. But the endoplasmic reticulum ribosomes, it takes them to the endoplasmic reticulum. And the translation takes place inside the endoplasmic reticulum. And then the protein which is formed, it is then processed, some methyl group, galactyl group, different groups are attached. So this is essentially, this is what happens in eukaryotes. So this is the whole picture of what happens. Now I will stop this whiteboard and I will show it. I'll just explain the note which I had. So these mRNAs which come out, they may have a sequence. This is a signal sequence present. This is recognized by a protein known as signal recognition protein. It comes and binds over here and it takes this mRNA to the ribosome. ঠিক আছে? To the endoplasmic reticulum, takes this particular thing to the endoplasmic reticulum, and over here ribosomes bind. Yes. So these are ribosomal, endoplasmic reticular ribosomes. And pre-cytosolic ribosomes, they are moving about in the cytoplasm. Moment mRNA comes, there is no signal peptide, they are translated over here. So essentially the ribosome is brought over to the endoplasmic reticulum. Therefore it is the rough endoplasmic reticulum. It looks very rough. because lots of ribosomes are attached on top. দাগড়া 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 হয়ে থাকে, ঠিক আছে? So after that, I'll just stop this and I will again share. Now let's see if I've opened that note. পাঠিয়েছি বোধহয় নোটটা, পাঠিয়ে দিয়েছি। হ্যাঁ, এটা পিডিএফ পিকচার পাঠিয়ে দেবো। এটা তো নোট নয়, এটা তো পড়ানো। রেকর্ডিং তো থাকেই, অল সব রেকর্ডিং রয়েছে, all recordings are there. You can, whenever you want, you can go and check the recordings. এই জিনিসটাই তো রেকর্ডিং এর মধ্যেই রয়েছে, তো তাই জন্য আমি পাঠাতাম না। ওটা পাঠাতে কি আছে, সব জাঙ্ক জমে যায়। এইগুলো দেখলে কোন পরিপ্রেক্ষিতে কোনটা বলেছি সেটা একটু অসুবিধা হতে পারে। যে এটা কেন বলেছিল, এটা অদ্ভুত একটা কিছু লিখে রেখেছি ওখানে, এইসব এরার্স ফেরার্স থাকে তো, মানুষ তো। তো এটাকে এখনো শেয়ার করিনি আমি এদের সঙ্গে যারা অনলাইন আছে। So now let's share this. Those who are online, can you see this?
Yes, sir.
Yes, sir.
ও বলছে না ওখান থেকে, talking about it. ওই যে এখান থেকে বাড়ি, ওখান থেকে আওয়াজ আসে। So here I've given the definition of what a gene expression is. এইটা পড়লেই যথেষ্ট। If you study this, this is sufficient. So it is the process by which a gene, it is used to direct the assembly of a protein molecule. We have seen how it does. The cell reads the sequence of the gene in groups of 3 bases, and this is known as read as triplets. Each group of 3 bases, they are known as codons, triplet codons. It corresponds to one of 20 different amino acids. So here let me tell, there are 20 standard amino acids which we have read. And we know that it is read as triplets. 3, read as 3. Now, how many types of combination for 20 amino acids? Say for example, if instead of triplet, it was read as a single nucleotide. If single nucleotide was read, you have A, T, G, and C. You could get only 4 types of proteins, right? 4 types of amino acids. Not possible. But there are 20. So it is not possible. If read as doublet, double, how many combinations can be, like AT or GT, read as doublets? That means it will be 4 to the power 2. It's read as double. The 4 codes raised to the power 2. कितना combinations हो सकता है? 2 अगर read होगा तो 8? No, not 8, 16. You have 16 amino acids, right? Still, what happened? 4 is 4. You have no explanation for 4 amino acids. So chances are, if it is read as triplet, then the combinations are 4 to the power 3, and that is, it can be 64 amino acids. Quite possible, but 64 is not seen. Only 20 are seen. 64 not seen. Now it was found that several codes may bring different amino acids. Just hold on. So if I was over here, so several different amino acid. That is, this means this genetic code which we are talking about, it is redundant. For example, it can very well be one amino acid may have two or more codes. Two or more different triplets can code for one amino acid. May have two or more codes. So now let us go look at this picture again over here. Ribosomes coming and binding to the mRNA. Those who are online, can you see it?
Yes, sir.
Yes, sir.
Okay, okay. So just enlarging this picture, this region over here. So you see it's read as codes, codons, U, G, C, because there is no T in RNA, therefore U. Then C, A, G, G, A, C. Each of them there is a code. So each particular code is bringing in via a molecule known as tRNA, transfer RNA. It brings the amino acid to this particular code. So there is a molecule known as tRNA or transfer RNA. So it has got a very strange shape. Clover leaf structure like this. This is the OH side. So here, say a specific amino acid is attached. A specific amino acid is attached to this RNA. And here, just complementary to the codon and anticodon, the specific amino acid codon is present. It is known as anticodon. So if this is amino acid, say for example, is methionine, it has got a code of methionine. Methionine, it has got a code of AUG or ATG in DNA. So the anticodon, this is codon. Anticodon will have what is the opposite of A? U, A, C. This will be the anticodon. So this mRNA, if it has got an anticodon of say UAC, something like this, always there is an enzyme which will attach a methionine over here. So there are 20 types of 20 amino acids, 20 types of the enzymes which will add 20 amino acids over here. This is the only case of methionine. If it's for proline, it will be another one, another enzyme. So that those are present. And this will go and it will go and sit in its particular codon. And there will be a codon anticodon binding. And what will happen? The amino acid which was attached, for example, C, this amino acid which was attached over here, it will go and it will sit, it will start elongating this amino acid chain and it will form the protein. This is how it goes. And the ribosome will move to the next codon. Here you see anticodon is binding over here and it will go to the next codon. Okay. K go. Moves to the next codon. So this is anticodon codon, and then once this is transferred, this tRNA becomes empty and it is released out. And since it's written triplet, the ribosome will move 3, 3, 3. The ribosome will keep on moving 3, 3, and here 1 amino acid, 2 amino acid, 3 amino acid, 4 amino acid will keep on adding, and this will keep on moving 3, 3. Each move, each codon, another amino acid comes, then it will go to the next codon, another type of amino acid will come till it finishes the end. A stop codon will come. This is actually what happens throughout. This is even much more enlarged picture. You have the growing polypeptide chain or the protein chain, growing protein chain, and it is attached to a tRNA. This empty tRNA is removed. This is the empty. This one has got the amino acid and tRNA is coming and binding over here. Then the ribosome moves here. This empty one is removed over from here. This is just an overview of translation. I'm not going in details of translation. I've got better pictures to show. So if you look at the thing of transcription, the enzyme is RNA polymerase, this brown color. So this is a DNA. This DNA is unwound. खुल गया DNA. And this RNA polymerase will attach these nucleotide triphosphates. That energy is utilized and complementary. A के opposite U, G के opposite C करके this starts to build, right? And like this, a mRNA chain will come and then a stop signal will come. This is how transcription takes place. And you have the translation. This mRNA comes out from the nucleus, ribosome attaches, and I've seen that this tRNA with amino acid is here. tRNA with amino acid comes and binds, এটাকে খালি করে দিল, and this polypeptide chain keeps on growing. So it depends on the different type of tRNA. It's known as also the transfer RNA. It is an adapter between protein and nucleic acid. This tRNA. On this RNA is attached the amino acid, and that amino acid comes and binds to the mRNA. Amino acid tRNA comes and binds to the mRNA, and this amino acid chain is transferred to the growing chain. That growing chain keeps on elongating and elongating, লম্বা হয়ে যাবে, হ্যাঁ, আর এদিকে এক একটা tRNA খালি হয়ে যাবে. Again this tRNA is again recharged in the cytoplasm. It is again recharged, and that enzyme comes and puts in another amino acid, যেটা খালি হয়ে গেল সেটা আবার ভরে দিল. The refill which was empty, that refill was filled up again. Again that refill will be used to write the new protein. So this is actually all these are the fundamental terms of gene expression. Next day we will talk, possibly day after tomorrow or on Sunday পরের দিকে, তোমার সকালে নয়, সন্ধ্যের দিকে maybe we will talk about lacoperon. Yes, lacoperon and gene expression. And then after that, following the next day, we will talk about online হবে Thursday তে এই টাইমই অফলাইন থাকুক, ঠিক আছে? বাকি দিনগুলোতে let's go online. So this is how actually I don't want to save it. Okay, do you people have got any questions? এই যে Ruchi, Shreyas, any questions? Abha? No, Shreyas says no,
No,
sir.
sir.
Okay.
No, sir.
Okay, then I'm stopping the recording then.
Okay, sir.
Okay, sir.
Okay.